Hi, my name is Chris Hardy, and I'm interested in studying obesity because it's one of the largest healthcare issues that we face today, and it's a growing problem. In just the last 30 years, the rates of obesity around the world have nearly doubled, and that's a huge problem because obesity is very closely associated to things like diabetes and heart disease, which ultimately lead to a shorter lifespan. And the tendency of westernized societies to eat more and exercise less is obviously an obstacle to treatment. But science has shown that there's more to obesity than just what you eat and how often you go to the gym. There's a clear genetic component, and understanding what genes make you fat and how that fat affects the heart is the main focus of my research. In order to address these questions, I use a very important model organism, the fruit fly, Drosophila melanogaster. And when I tell people that I study really fat fruit flies, they usually think I'm crazy. But flies are much more similar to humans than people might expect, with about 75% of genes that cause a disease in humans also found in the fruit fly. You can even take fruit fly insulin and activate the mammalian insulin receptor, and vice versa. So here at UNLV, we've established a very unique system in order to study obesity in fruit flies. It started about 15 years ago when my advisor, Dr. Alan Gibbs, took a set of fruit flies from the wild and started a long-term evolution experiment in the lab. Each generation, he would select for only the flies that could survive the longest without food, and these tended to be the ones that could store the most fat. So over time, it became survival of the fattest, as fat genes were passed down from generation to generation, to the point where now, some 95 generations later, we've evolved fruit flies that are extremely obese, so fat that they even have a hard time trying to fly. And so my first question was to understand if they also suffered from heart disease. And they do. We took advantage of a machine that optometrists use to look at nerve structures in the eye, and we turned it around to look at heart function in a fly. And here are the results. You can see in a normal skinny fly, the heart is fully open, completely contracts, opens and contracts rhythmically over time. In our evolved obese lines, the heart is wider, it's dilated, and it's not quite as contractile. It's a condition known as dilated cardiomyopathy, and it's similar to the kind of heart disease you would see in obese humans. We found that this was largely caused by a large amount of fat tissue that physically altered the normal placement of the heart. We zoomed in on the fat tissue into the individual cells, and we found that they contained extremely large lipid droplets, and these are the compartments in the cell that hold most of the fat. What's really cool is that the genes that control lipid droplets, their size and how much fat they store, are very well conserved between humans and flies. So the next step of the project was to try to figure out what genes are causing this. So to do this, we performed a genome-wide association study where we took DNA from a skinny fly, DNA from a fat fly, and we sequenced it. So now we know all the A's, T's, G's, and C's that make up a skinny versus fat fly. Doing this, we were able to find many, many genetic differences across their genomes. And we tracked the most important using a complex computer program that we developed. We were able to use these locations to identify a set of 1,500 genes that were associated with obesity in our flies, and 1,000 of those were transferred over to humans, or they have a human equivalent. And these genes were associated with muscle disease, heart disease, and type 2 diabetes. So now we can go through this list, start testing them, and some will be targets for therapeutic intervention in the future. Thank you.